I'm sure some of you may have, you can find them afterwards. We'll move right into Nick Sard from SUNY Oswego to talk about parentage-based tagging for Lake Ontario. Chinook salmon, insights and future discussion.
Sounds good. Thanks. All right, hey everybody. Looking forward to talking about parentage-based tagging, or PBT for short. Just want to acknowledge my collaborator. So Vinny Siegel, my lab manager, is the one who's really in the lab processing all these samples. I've got an undergrad pictured here on the PowerPoint, Eli, who, when I talk about the sex marker stuff, and you'll know about that in a second, that was really work that he's been doing for his honors thesis, and Grace Cordone is a technician in the lab that helps process some of these samples. And I'd also like to thank Mike Connerton and Chris Lagarde in terms of supporting this PBT work. And Steve, when he started this pelagic session, and Mike hit on this too, one of the things for this Chinook salmon alewife predator-prey relationship is to understand how much wild production we have in the system through time. And that's the big uncertainty. This relative contribution of wild Chinook salmon to the lake-wide population is uncertain. Mike had put out an estimate of 50%. That's now a little bit over a decade old, and the quest is to get a more regular estimate of this to understand how that varies through space and time in Lake Ontario, so we can decide how many hatchery fish to put into the lake to maintain this predator-prey balance. That is, we know that alewife make up the majority of the prey base out in the lake. We know that the majority of the food that Chinook eat is alewife, and so it's really important that we get that number, get a good estimate of that number, so we don't allow the Chinook to eat themselves out of house and home. This work was actually started by Kimberly Fitzpatrick, a past PhD student at Cornell University from Suresh Sethi's lab. Both Kimberly and Suresh have since left Cornell. Kimberly is a postdoc at the University of Alaska Fairbanks, I believe, and Suresh works down at the University of Brooklyn College now. The other co-authors on there, Nina and Harmony, I believe are still at Cornell but aren't involved in the project, and Ben Marcy-Quay now works at USGS. So long story short, the people that started it aren't there anymore. I have a fair amount of PBT experience. I did my PhD at Oregon State, and I studied Chinook salmon for 5 years in the early 2010s. Long story short, so I'm well positioned to work with Chinook salmon and use this technique. So I recently took over the project, got my first set of samples from Mike last summer, and we've been starting to process those we're working towards generating these reconstructed pedigrees to get estimates of wild production. The idea with parentage-based tagging, this new tool, first off, I wanted to say thanks to Stacy Fergal, New York State Sea Grant, for working with a local artist to create this really nice graphic that visualizes the parentage-based tagging process. The parentage-based tagging process, how good is the... So, let's see here. I get two screens to choose from. This is always interesting. I'll start over here. So there's a simplified version, A through F. I'm going to go through here and kind of talk about this circle. So what we do is we collect samples from all female parents that are used in hatchery, used as hatchery brood stock. So we take those tissue samples and we can process them, we can genotype them, we can get their DNA fingerprint, and we can put that into a database here. Their offspring are then released, so these are hatchery origin offspring. They are released into the wild at some point. Along the way, wild origin offspring are also produced in other parts of the system, and they are also moving out into the lake themselves. In the lake, the population is a mixture of hatchery and wild fish. These fish are caught by charter captains and charter vessels and their crews, as well as just anglers in the lake itself, and these fish can be sampled, that is, tissue samples can be collected, scales can be collected for age, and these fish can also be genotyped. and then we can compare these genotypes from the fish caught in the open lake fishery to the brood mothers that could have potentially produced them, to determine which are hatchery fish. Now, we're interested in wild fish, so it's the fish that don't assign that are the ones that were most, well, it's two sides of the same coin, right? But the wild fish is the thing we're really after. The annual sampling structure is that we sample about 1,000 broodstock mothers. Those come from the Credit River and from the Salmon River each year. There's a set of validation samples that I'll get into in a second. But then there's a set of open lake fishery samples. And like I said, they're collected from a set of charter anglers, or as well as from creel surveys, so DEC personnel collecting fin clips from fish that are brought back to the harbors. And the idea is that for the brood mothers can assign back to age 1 through age 4 fish, and the majority of those fish should be age 2 or age 3 fish. That's the expectation. We have brood mothers that go back to 2018, and we're waiting for brood mothers. So there's kind of like this gap where Kimberly moved on from her PhD, and I took on this project, and so there's a set of samples that are actually being processed by an external group led by Nate Campbell, a guy who created the technique that we're actually using called GT-seq. So we're still waiting for those data to come in. The 24th is the date he says they'll come in. We'll see if that's true. And I've got about 30 libraries. So that 30 libraries, what that means in genetic speak is that's about 3,000 fish are ready to be genotyped. We're just about ready to send those to Cornell. So I told you that I would talk about these validation samples. So the validation samples are trying to get at testing how well our method works. And so to do that, we have a set of roughly 100 known hatchery fish that come right out of the hatchery, right from the brood mothers, and we try to assign them to the brood mothers. Expectation is we should assign every single one of them to the brood mothers. And when we don't, this is an estimate of our false negative rate. So the number of fish that should have assigned as a hatchery fish but are interpreted as a wild fish. And then we also have wild fish. So these wild fish, these are age 0 fish caught by New York State DEC, where we're sure that there aren't any hatchery offspring. And so there's about 100 of those, and the same type of thing. We try to assign our brood mothers to these wild offspring with the expectation that none should assign. And so if none assign, that's great, but if we do have some assignment, then that's an estimate of our false positive rate. And these two rates can be used to get a better understanding of when we go out to the open lake fishery and we make our assignments and we get at some number of those 800 fish being assigned as hatchery offspring and some are assigned as wild offspring, we can account for the error rates that we're observing when we compare to these kind of known sets of samples. And you can see, this is a table out of the original publication that Kimberly wrote, that there's about 95% of all known hatchery fish in columns assigned a hatchery, known origin fish in columns. We have hatchery origin on the left, wild origin on the right, and then in terms of the rows, that's assigned as a hatchery fish or assigned as a wild fish. So on the diagonal here, we've got 95% assignment rate for hatchery fish being assigned as hatchery fish, so that's our false negative rates. These are falsely assigned as wild fish here, there's about 5.5%. And then for the wild origin fish, these are known wild fish that shouldn't assign to our hatchery mothers. 98% of them do not assign, but we did get one assignment. So we being Kimberly and her colleagues. And so that practice is going to be implemented on an annual basis to get an estimate of false positive and false negative rates, along with some other ideas I have. So from my perspective, what happens in the lab is that we get tissue samples from New York State DEC, from OMNR, and we isolate genomic DNA from females. I've got females underlined there because the whole idea, as Kimberly wrote in her title, is trying to balance accuracy with cost. It costs money to process these samples. There could be 3,000 fish to genotype on an annual basis for broodstock if you did both moms and dads. They do a 1-to-2 breeding design. So the idea is focus on the moms and just assign the moms to the kids, and that should be sufficient. But the kicker is that you got to know that which ones are females, and so I'll get into that in a second. Then once we've isolated DNA, we can genotype these fish at what Kimberly and colleagues identified as 61 microsatellite loci that can be genotyped in parallel using this common technique in next-generation sequencing called GTC. Once you have genotypes for all these fish, you can reconstruct the pedigrees using the mom's genotypes and the potential kids' genotypes. We'll be doing it with multiple programs and comparing them. The ones that I have up here are Colony and CKMR-SIM. These are common and well-established methods. So we're currently working through, my lab is currently working through kind of 2 logistical challenges. And I'm going to go through each one of these and spend a little bit of time. One is some hatchery samples were sent to us with a mixture of males and females in the same vial. So I can't tell which one's a male, which one's a female by just looking at the fin clip, and that's a challenge for us, and I'll get into that in a second. And the other vague statement I have in here is considering pedigree model assumptions. So we're going to get into the weeds a little bit with that one as well. So I've got that logistical challenge number 1 up there as my title, highlighting that there's males and females in the same vial. And our objective is to determine males. So Mike said this to me. I said, well, that's no problem. I did this during my PhD. Like one of my first publication, we used a genetic-based method to sex the fish, and it was pretty good. It was 90% concordant with phenotypic sex, and then the discordance was largely explained by chromed out fish that were caught early in the season, and they couldn't differentiate males and females. So I think it should do a pretty good job in Lake Ontario. And the assay is really simple. It was developed in 2008 by a group out in Washington, Brunelli et al. And the assay is really, you can see it here in the gel. I'll go over here because I'm neglecting you guys. I guess I can't because the mic's over here and I know someone will yell at me. Sorry. So I got four columns here: female, male, female, male. The females all have this bottom band, and then the males will have a larger band. So we're looking at a gel here. We're looking at DNA fragments of specific sizes, and using these banding patterns of the gel, you can clearly differentiate males and females. Great. Sounds simple, right? So what do we get when we look at salmon river fish? So salmon river fish, we had 600 all known females. Okay, and so this is up here. This top gel is all salmon river fish, and our ladder is kind of, it's not, you can't see it very well here, but I have the known sets of DNA identified. This band is the female band. Okay, great. But this band up here should not be here, right? That's the male band. But these are all females in the salmon river that had eggs. They literally cut them open. You saw the eggs. You know that, and they produce offspring. So what's going on? I don't know. With a credit river fish, a very similar thing. We see this female band, but we see the vast majority of the credit river fish having a really strong male band there. And so the question is, what's going on? 2 hypotheses to explain these observations are that the marker O2Y3 does not reflect phenotypic sex of the Lake Ontario Chinook salmon population. I mean, it's a non-native fish. There's certainly a founding effect. There could be a chance that a mutation occurred and drifted to high frequency in the population. That's certainly possible. There's also a possibility of contamination. If you've ever been to a breeding event at a hatchery, it can be kind of a messy experience, but particularly when you have lots of fin clips in the same vial. Who knows? It could be contamination. And so we kind of thought about, well, how can we test these hypotheses? So one is that, well, this marker is kind of old school. It was produced in 2008. It works for lots of different populations in the Pacific Northwest, but there's a more modern marker out there. So I put a call out to my colleagues on the West Coast and said, hey, do you got a better marker for me to use? And they came back with, yes, we've got one called OTS_sexy31. So I said, sounds good, I'll use that. And I got the same result as OTY3. So, now what? If you look at where these markers are in the genome, because the Chinook genome is published, OTY3 is actually within 1,000 base pairs, i.e. very, very, very close to the SCY gene, the sex-determining gene for Chinook salmon. So you would expect OTY3 to actually do better than OTS-sexy31. And so we're trying to figure out what the next steps are. One, Lake Ontario is not the only population where there's discordance between genotypic and phenotypic sex. I've got 3 studies up there on the left, and that highlight that that's true. There's some more recent studies that have identified non-functional SCY genes in some populations, so we're going after that. I'm going to speed through this and just say we are looking at the genetic material contamination hypothesis using traditional methods. So traditional methods, you can clearly see when there's more than 2 alleles. So on the x-axis is size of the marker, and on the y-axis is relative fluorescence. But the point is you're only supposed to see 1 or 2 peaks there. And when you see more than 2 peaks, then that's an evidence of more than one marker amplifying or contamination. So we're trying to ground truth what we're seeing with the sex marker using traditional microsatellite genotyping. And then the logistical challenge number 2, because I'm basically out of time, I'll just say the number of alleles observed per locus is good. The percent fish genotyped is not bad. As a professor, I'd give it a B, maybe a B minus. We could do better. Most of the pedigree projects that I ever worked on, more than 95% of all the fish are genotyped at all the markers. And then there's this measure FIS. So I saved you a population genetics lesson by talking too much. But generally, the idea is that this is a proportional deviation from Hardy-Weinberg expectations, and it should be near 0. So what this says is that the vast majority of the markers, this box plot on the far right, are about 20 to 30% off compared to what we'd expect from Hardy-Weinberg expectations, and that's generally not good, in short. So yeah, at that I'll just say we're continuing the sex marker investigation. We're going to do some simulations to look at the performance of the marker set that we have. GTSeq's nearly done processing their samples. I already told you about my libraries, and I'm looking forward to reconstructing some pedigrees in the near future. Be happy to take any questions if I have time. No, sorry.